In this video, we will discuss about specificity and efficacies of chemogenetic method. So we will continue our discussions about the chemogenetic methods. That is the two methods that they are mainly used. First one is this RTX, and second one is through this HM4DGI. And both of these methods are tested. Which of them can able to give better tumor suppressions? So in the panel F, what they have tested these methods in the absence of slit or slit knockout samples, slit knockout B16F10 cells. So what they have able to find out that in the normal case, they are able to see tumor suppressions for both the methods, that is through the RTX and through the HM4DG. We can see that. And however, when the slit 2 has been knocked down, they are not able to see changes compared to the control. The changes are non-significant. And then when they reintroduced this recombinant slit 2 in this knockout cells, they are able to see a tumor suppressions again, or the tumor suppressions properties are been regained by the system. This indicates the role of the slit 2 towards this tumor suppressions. Sorry. And similar things they did it with ATF4 knockdown. In the control, they are able to see tumor suppressions for both the methods, but when the ATF4 is knocked down, this tumor suppressions cannot be seen compared to the control, and the difference are non-significant, except for the case of this HF4DGI, where tumors are actually increased. And in the third case, or in this case also, when this recombinant slit 2 are been reintroduced in this ATF4 knockdown B16F10 cells, they can see that tumor suppressions abilities are again regained by both the system. So this figure F and G indicates the importance of this slit 2 and ATF4 in the process. Thereafter, they checked some of the negative control to check the specificities. So in the panel H, they checked whether since this DCG is required in order to trigger this chemogenetic method using HM4DG. So they first checked in the non-optogenetic conditions with normal drinking water and the DCG drinking water. They do not see any tumor changes, indicating the DCG alone does not cause any tumor change. And thereafter, they also checked it on the wild type and wild type with light or optogenetics. In that case also, they do not see any significant changes. And afterwards, they checked it for some more different conditions where they use different transduced cells, and in that case, they do not have this catch triggered plasmid, that are catch enzymes that are, or the catch proteins that are usually upregulated in the presence of light. Those protein is not been transduced. Those protein sequences are not been transduced. So because of that, it is not been triggered, and there is no tumor volume change significantly among all the cases. This indicates the specificities of the method. Afterwards, they wanted to find out the role of CGRP. Since CGRP also shows up previously that it has a direct role towards tumor progressions, so they checked it in the case when the positive control where the tumor's volume is gone up, where they do not have HM4DGI. In that case also, they are able to see the high amount of CGRP, whereas when they use the HM4DGI to tumor suppression, they see reductions in the CGRP content, indicating CGRP has a direct role towards these formations, towards this tumor development. Then in the mouse model, what they did is they knocked down this Calca gene, and then confirm the knockdown of Calca gene in this mice model. Calca gene knocked down to some extent, but those decrease is statistically significant. It can be seen here. All the knockdown genes gives EGFP signals, whereas a knockdown gives no fluorescence signals. So from that, they are able to distinguish how many genes have been knocked down, and they say that there is a significant difference or significant decrease in the Calca gene, but those difference is not very high. So however, when they check the effect on the tumor volume, so in the case where Calca gene is been knocked down, they do not see a decrease in the tumor volume compared to the case where there is no HM4 DGI is used. However, when they regionally administered the CGRP, they are able to see increase in the CGR tumor volumes when there is no reagent, that is HM4 DGI, to activate the this tumor suppressions mechanism. However, even in the case of where HM4 DGI is present, they are able to see a increase in the tumor volume compared to the case where there is no CGRP. High CGRP cells shows more increased tumor volume because CGRP's upregulation is directly related to the tumor progressions. So because of artificially injection of CGRP cause more tumor growth compared to the control case. This is how they checked. So this is how they checked the specificities and the efficacies of the chemogenetic method. So if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.